Hi everyone, in this lesson we're going to be looking at how proteins are newly made, and we're going to be focusing on the first stage of that process, which is called transcription. These are the key words that you're going to have to use when answering questions about transcription. Please make sure that you've read the pages in your textbook and that you are familiar with these terms. The learning objectives associated with today's lesson are to compare the structure of DNA with another similar molecule called RNA, and understand that there are three types of RNA which are involved in protein synthesis, and those are mRNA, tRNA, and rRNA. While we're explaining the process of transcription, you need to be able to include the following terms into your writing of your answers, and those are helicase, RNA polymerase, template strand, nuclear pore, triplet, and coding strand. So we're going to just step back from looking at that process for a second and look at what a gene is. Now here in this little cartoon diagram, we've got a picture of a single chromosome with sections highlighted. So a gene is a section of DNA, and we know that DNA is wound up into chromosomes, so it's a section of a chromosome and DNA, and it forms a sequence of nucleotide bases. So this small section here, if we look at it, if we zoom in on that DNA strand, we can see that there are nucleotide bases there in a particular sequence. Now that particular sequence will code for one or more proteins, and those proteins can be passed on to future generations. So we describe them as units of heredity. Now these genes and this coding sequence can code for specific traits, such as brown eyes in this case, or hazel hair. It could be blue eyes, blonde hair, and so on. Now the majority of the genes, as we know, are found inside the nucleus of the cell. However, you do get about 37 genes that are found in a circular disc in the mitochondria, and that forms mitochondrial DNA. Now the specific location where these genes are found are called the locus. So if we're talking about more than one, we're talking about loci. So the locus for the gene for eye color is found on the upper portion of this chromosome. Before we go any further, it's important that we understand the differences between DNA and a similar molecule called RNA. So RNA, or ribonucleic acid, is just simply another type of nucleic acid that's found in the human body. It is very similar to DNA, but there are specific differences. So what I'd like you to do is to pause the video here and read those pages in your textbook and answer questions 1 to 3 on pages 7 and 8 of your workbook. Alright, so hopefully you've gone through those pages in your booklet and we can review your knowledge. Now here you can see an RNA nucleotide, and it's really similar to DNA. We've got that phosphate group at the top, and we've got this pentagon in the middle, and that is still a sugar. It's a pentose sugar, but this time we have an additional oxygen, which we can see down here, and hence it's ribose sugar instead of deoxyribose sugar. Another key difference is that although we still have 4 nitrogenous bases, there is a notable absence of thymine, and instead RNA has uracil. So just to recap, RNA is a type of nucleic acid. It is made up of repeating units called nucleotides, which have a phosphate group, a ribose sugar, and one of 4 different bases. Now, unlike DNA, RNA is single-stranded, and the complementary partner to adenine is going to be uracil. And here in this diagram, we can see that although it's single-stranded, it's very similar to DNA in the fact that it winds up into a helical structure, but in this case, it's a single helix instead of the double helix that we see with DNA. So now we're going to be looking at the 3 different types of RNA that we find. The first one we're looking at is rRNA, or ribosomal RNA. You can see in the little cartoon diagram here, this whole structure is the ribosome, and the ribosomal RNA is found in the lower portion. So about 60% of the mass of ribosomes is that RNA, with 40% being protein, so it's quite significant. Now, this ribosomal RNA ensures that our mRNA and tRNA molecules, which we will talk about later, line up correctly. Another role that our RNA plays is it has an enzymatic role, so it speeds up the rate of reaction in forming peptide bonds between adjacent amino acids. Now, as we unpack the process of protein synthesis, this will become more familiar to you, and you'll understand what the function of our RNA is a little bit better. Right, so the next molecule we're going to be looking at is transfer RNA. Now, transfer RNA is this shape here, this rectangle, and you can see that it has an amino acid attached onto the top. Now, transfer RNA is really important in protein synthesis. It carries amino acids to a section of code, and it places them in the correct order. And the final type of RNA we're going to be looking at is messenger, or mRNA. Now, messenger RNA is found inside the nucleus and outside the nucleus, but it's actually manufactured inside the nucleus. What mRNA does is it reads this DNA code and it makes a copy. So if we think of DNA being the instruction manual for making proteins, DNA can't leave the nucleus, so we have to get that instruction out of the nucleus somehow. So what we do is we make a copy of it. So mRNA will copy the particular code for a gene, because remember, genes code for proteins, and it takes it to the cytoplasm, where that code is then made sense of or translated into a functional protein. So now that we understand a little bit about the differences between RNA and DNA, and we understand that there's three types of RNA, which are tRNA, mRNA, and rRNA, you should be able to complete and check your answers to the questions on pages 7 and 8 of your booklet. Here is a summary of the differences between RNA and DNA. Please make sure that you have a note of these and that you understand that there are key differences that you will be asked to write about, particularly in compare and contrast questions. Okay, so now we're going to be looking at protein synthesis. Now, this comes in two stages. The first stage we're going to focus on today is stage 1, which is transcription. So the first stage is when the code to make a new protein, so that gene sequence, is copied from DNA, remembering that DNA cannot leave the nucleus, and it's taken out of the nucleus to the cytoplasm and then eventually to ribosomes. So the second part of protein synthesis is when this instruction code, copied by mRNA, is then used to build new proteins. So before we talk about transcription and translation in more detail, we just need to cover some of the key terminology that we need to understand. So start off by looking at this little cartoon here. The blue at the top is a section of a gene. So we can see the backbone of DNA and we can see the nucleotide bases. Now, when we copy a section of a gene, we copy it in groups of 3. So the first group of 3 in DNA is called a triplet. And the corresponding group of 3 bases found in mRNA is known as a codon. Now we give them different names so we know where they come from. So in DNA, a group of 3 bases is a triplet. A group of 3 bases in mRNA is known as a codon. And you can see here that the bases are complementary to each other. Now eventually, the group of 3 bases from that triplet and then that codon will be translated into a single amino acid. And the way that these amino acids join together in that particular order will make a protein molecule. And this is why DNA codes for proteins. All right, so now we're going to start looking at the process of transcription. So remember, all of this happens inside the nucleus of a cell. First thing we're going to be looking at is the fact that in order to copy a set of instructions, we need to look at the source code. So the source code is a section of DNA that codes for a particular gene. Now, in order to read that gene sequence, the DNA must be unwound and unzipped, which we can see here in this little section. So the enzyme that unwinds and unzips that particular section of DNA is DNA helicase. The unwound and unzipped portion of DNA that we are going to read is called the template, the DNA template. Now, RNA needs to know where to start copying from. So just like in DNA replication, transcription uses a similar sequence of events. We have a promoter sequence, which you can see in pink here, and that promoter sequence tells RNA molecules where to start copying from. So the enzyme RNA polymerase, which is this orange thing in the background, this will bind to the promoter and it will start copying from that position. So this is the start of transcription. The next section, if we look here, we can see that the section of DNA that's opened up, and the blue in this background in this cartoon is the RNA polymerase. This will catalyze the reaction of free RNA nucleotides binding to that template strand in DNA. Now, we only need to copy one side of DNA because if we know one side, we know the other side because they're complementary to each other. So these free nucleotides will come along and bind to the relative nucleotide on the DNA and form hydrogen bonds. Remember, hydrogen bonds are formed first because they are weaker. Eventually, you get covalent bonds forming between the sugar and the phosphate, and that becomes the backbone. Eventually, this process continues until we reach the end of the coding sequence, and as a result of that, we have a brand new mRNA molecule. So this cartoon diagram is just to show you in simple terms what's happening. So here we have our DNA molecule, and DNA helicase has unwound and unzipped the portion of DNA that codes for our particular gene sequence that we want to copy. And we can see here in this zoomed in region, this is our template strand, this brown strand on the top, and we can see the nucleotide bases. What starts to happen is free mRNA nucleotides will start to line up with their complementary base partner. So here we can see uracil combines with adenine on DNA. These light green lines are showing that we have weak hydrogen bonds forming between the complementary bases, and the backbone here is a covalent bond that forms between the sugar and the phosphates. So once we've got our finished mRNA molecule, it has to leave the nucleus. Okay, so remember, the nucleus is only where their DNA is contained. The organelles to build proteins are out in the cytoplasm. So our newly formed single-stranded mRNA molecule will leave the nucleus via a nuclear pore. That's a gap in the nuclear membrane. And from there, the DNA molecule will rewind and rezip and condense back down into the structure that it was before. Okay, so here is a diagram showing everything we've talked about so far. Again, I know it looks busy, but let's just take it one step at a time. At the top here, we can see that the DNA molecule is unwinding and unzipping, exposing those nucleotides. We have a promoter sequence shown in pink here, which tells RNA where to start coding from. As RNA polymerase moves along that molecule, it promotes the free RNA nucleotides to add to that gene sequence in a complementary way. So we have RNA nucleotides binding, forming hydrogen bonds with the DNA template, while the covalent bonds are forming along the backbone. Once we have that completed backbone forming, the hydrogen bonds between the RNA and the DNA break, and eventually we have a completed mRNA strand, which will then leave the nucleus via a nuclear pore. Okay, so what I would like you to do at this point is to pause the video here and see if you can write a summary of what's happened. Now, please make sure that you use your notes in your textbook to help you. This diagram may also help you to write a summary. You can use this as the basis of a labeled diagram if you wish. Okay, so now that you've completed your own summary of transcription, let's review what we've learned so far. First of all, we know that DNA codes for specific protein sequences, and those specific codes are known as genes. We know that a group of 3 bases in DNA is known as a triplet, while a group of 3 bases in mRNA is known as a codon. The first stage of protein synthesis is known as transcription, and this is where the gene sequence from DNA is copied by mRNA. mRNA is a single-stranded sequence that is copied from DNA. Once this is fully copied, it will leave the nucleus via a nuclear pore and enter the cytoplasm. And we also know that there are key differences between DNA and RNA. We know that RNA has uracil instead of thymine. We know that RNA is single-stranded. We also know that mRNA can leave the nucleus, and RNA molecules in general have an extra oxygen. Okay, so now that we've covered transcription, I'm just going to go through a really basic animation to summarize that with you again. So here we've got a diagram or a little cartoon showing DNA. We know that it's DNA because it's double-stranded, and we have thymine instead of uracil. So if we look at our section of DNA side on, this is just a really basic diagram showing the nucleotides in the backbone. We can see that the DNA will start to unwind and unzip, and the bases will break hydrogen bonds, and that reveals the nucleotide sequence. When that happens, the sense and antisense DNA strands are exposed. So the sense strand is the one that we copy, the antisense strand is the one that goes in the anti-parallel 3 to 5' direction. Here we can see the RNA polymerase molecule attaching to the start where a promoter would be, and it moves along the DNA molecule, reading that code, and it promotes mRNA nucleotides to start combining in the correct order so that they combine with their complementary partner on DNA. So as that mRNA molecule is synthesized, RNA polymerase moves along the chain. Eventually, the RNA polymerase enzyme will stop, and this will send a signal to the mRNA nucleotides to stop being added to the gene sequence. At that point, RNA polymerase will detach and move away. And what will happen next, as you can see here, is the hydrogen bonds anchoring the adjacent nucleotides together will break, and that mRNA molecule will start to move out of the nucleus, and the double-stranded DNA will start to rezip and rewind. Okay, and we can see our mRNA molecule moving out of the nucleus via a nuclear pore, and that moves to the cytoplasm where it will bind to a ribosome. Now, if you're still unsure about the process of transcription, this is a really good video to watch. Just watch the first part of it, so that's up to 6 minutes 50, and that will give you another alternative overview of what we've discussed. Now, before we finish up, have a go at the questions here. So, first of all, what type of nucleic acid is shown in this diagram? Question 2, what type of bond does this arrow point to? And what base is missing from RNA that is present in DNA? Question 4, what base is that substituted with? Question 5, where does the first stage of protein synthesis take place? And question 6, what is the function of RNA polymerase? Okay, hopefully you were able to answer those questions, and these are the answers. If you got any of those wrong, I would really suggest that you go back and review your knowledge and read the textbook to ensure that you understand all of that terminology. Okay, so just to recap, what I would like you to do is to complete questions 1 to 11 on pages 8 and 9 of your work booklet. Please make sure that you understand the process of transcription, and please make sure that you read pages 235 to 236 of your textbook.